In this lab, we will talk about respiration and photosynthesis, and we will focus in this year mainly in photosynthesis. So in this process, and we are going to try to understand what is happening during photosynthesis. So the main idea is, if you watch the video, we're going to do something similar, but remember, as in all the labs, the videos there, they just want to show you more or less what we are doing. We are not exactly doing the same things. I just wanted to show you, in case that there was a closure, how you can do this experiment. So you're going to have a setup that allow you to have light. That is important for the photosynthesis. You're also going to have to prepare the disk. So we will have the leaves ready for you to do, and you are not going to exactly use this. You're going to use the similar cylindric metallic thing that you use for the potatoes, and you're going in that way. You're going to make holes, circles in the leaves of the spinach. And as I said, this is just an indication of what you have to do. We are going to do it slightly different things. So instead of using this light, we have this light if we are in person. If not, you might use something like that at home, and I will show you how to control it. So one person in the lab can use these two, while the other person can use the other one. And you can read this, is very helpful, and you can follow those instructions. This place the water in the petri dish is very important. So again, this is not going to be exactly like that, but the idea is, when you make the holes, make it in these sections as they show in the videos. Don't make it here. And every time that we talk about infiltration solution, we will give you an infiltration solution there, but we are going to do it slightly different this year. Instead of having an infiltration solution that you will have to swirl and you have to do this for a long time, what we're going to do it is using a syringe. So I show you this video, and it shows how to do it. Advice. Last year I thought that this was to remove the oxygen and the CO2. The strength was the most important, but no, it's not the strength. You can do it very gentle. The important is that you start removing the air from that. I already uploaded a video, and we will practice in the lab. And the idea is that when you remove the air, the disk will fall in the bottom, and due to photosynthesis, as they have to produce oxygen, so they will need to absorb CO2 and release oxygen. They will start floating. So remember, after you do this, they have to go on the floor on the bottom of the beaker. Then when they start doing photosynthesis, you're going to quantify how the time, how long, what is this take, 2 minutes, 5 minutes, 4 minutes. And you're going to have different solutions. So here you have your discs, and you're going to have a solution with bicarbonate. This is very important. You should have this on. You should have this in white light. You should have the maximum intensity. This is super important for the first experiment that you're going to do. For if you want that to work, remember, always maximum intensity. And as I said before, we are just going to evaluate how long this process takes to occur. If it occurs really fast, or it takes longer. And you are going to quantify 10 disks. In another part of the lab, we will try to test our hypothesis. So we will do the same experiment, but without light. So how you're going to do that? Remember this sacred place that you are not supposed to touch because it's from Janus and you are not supposed to show to contaminate that region. In this case, we're going to make an exception and we're going to put our experiment there. And in that way, there will be no light. And you close it, and after 20 minutes, after 10 minutes, you can open it to see if they start going up or no, the disk. So this could be a way to test this question: do light-dependent reactions require carbon dioxide or not? And to clean, there will be a compost beaker that you can get rid of the spinach. And remember to clean up by rinsing the beakers and rinse it 5 times as usually, and put it upside down when you're done.